Hello guys, welcome to my channel. So in this video I'm going to talk about the C4 cycle which connects with the Calvin cycle, which is being described in the earlier video. It is also called as the C3 cycle. So in C4 cycle, what is the difference between a C3 cycle and a C4 cycle? So let's discuss that. In a C4 cycle, a C4 plant is involved. A C4 plant is a plant that cycles carbon dioxide into a 4 carbon sugar compound, and that compound we're going to discuss it. So what are the fundamental properties of such type of plants which is being described as C4 plants? These plants are very efficient in hot dry climates and make a lot of energy. So this is the benefit of performing a C4 cycle. So the examples of such plants are like corn, pineapple and sugarcane, and many more plants are there. So in a C4 cycle, the plants are growing under dry hot environment. In high temperature, these plants are performing and producing a lot of energy. Such plants can photosynthesize even in low carbon dioxide concentration, even though there is a very low concentration of carbon dioxide, and also these plants are synthesizing ATP and also under the partial closure of stomata. Such plants do not require high amount of water content. They can easily activate their cycle even in low water content. High temperature is there, high light intensity is there, and photorespiration. As we all know that photorespiration is a highly wasteful process because RUBP oxidizes in the presence of oxygen. In the process of photorespiration, I am going to discuss this video in the next session. And in C4 cycle, the photosynthetic rate is very high. And now in C4 plants, there is some of the characteristics which are very necessary to read about them. The leaves of C4 plants have the presence of dimorphic chloroplasts. What do you understand by the dimorphic chloroplast is that there are two types of chloroplast. Even though there is a morphic term here, always remember the morphic term represents what? The morphic term represents change in forms, and it is called as the Kranz anatomy. This term is very much important. In these plants, the vascular bundles have a sheath of large parenchyma cells and which wrap around a rath, and it is called as the Kranz anatomy. So as studied earlier, they have dimorphic chloroplast, two types of chloroplast. In one sort of chloroplast, the mesophyll cells are smaller and have well developed grana called as the granule chloroplast, and in the bundle sheath cells, and in the second type of chloroplast, the bundle sheath cells are larger which lack grana. Grana are the stacks. Each grana consists of stacks of thylakoids. If you remember the structure of the chloroplast, so they lack grana but contains numerous starch grains. In C4 plants, the initial acceptor of CO2, as discussed earlier in the C3 cycle, as you all remember, if you have watched my video. If you haven't watched my video, please go through my channel. There is a video called as the dark reaction C3 cycle. Go through it. It is very much brief. You can easily remember it. In C3 cycle, the initial acceptor of carbon dioxide was RUBP, which is ribulose 1,5 bisphosphate. But here in C4 plants, the initial acceptor of carbon dioxide is phosphenol pyruvic acid or phosphenol pyruvate, which is a 3 carbon compound. And now it is most important thing to determine this reaction. CO2 has 1 carbon, PEP has 3 carbon, 1 plus 3 equals to 4. So it forms a C4 acid which is called as the oxaloacetic acid under the influence of an enzyme called as PEP carboxylase. In C3 cycle there was Rubisco. Here it is PEP carboxylase to form a C4 acid called as the oxaloacetic acid. So here also carbon fixation takes place and occurs in the mesophyll cells. And now if the oxaloacetic acid is the first stable product of this cycle, it degenerates. It goes to the bundle sheath cells. Here it is most important to discuss that C4 cycle, in C4 cycle it occurs in the mesophyll cells. In the mesophyll cells the C4 acid, which is OAA, oxaloacetic acid, is formed. This oxaloacetate moves from one chloroplast to another, from the mesophyll cells to the bundle sheath cells. In the bundle sheath cells the oxaloacetates undergoes decarboxylation. It releases one carbon dioxide, and that released carbon dioxide is used in the C3 cycle for the requirement of CO2. So decarboxylation occurs at bundle sheath cells, and again that compound which we have discussed, that OAA releases carbon dioxide, so it produces a 3 carbon compound. That same 3 carbon compound goes to the mesophyll cells and it regenerates into phosphenol pyruvate, so that again a C4 cycle can take place. Here you can see the structure, what I was talking about. PEP takes CO2, carboxylation occurs to form a 4 carbon compound, and this 4 carbon compound is called as OA. 4 carbon compound OA undergoes decarboxylation in the bundle sheath cells. It releases CO2 to produce a 3 carbon compound. This CO2 is utilized in the C3 cycle or Kelvin-Benson cycle, and this 3 carbon compound regenerates to form PEP, which is phosphenol pyruvate. So here it is, very much clear. Hope you like my video. Like, share and subscribe.